So, and just I would like to mention the collaborations within CIAT, especially the work package 2. So they did a lot, most of the work in the lab. Really appreciate their help and the collaborations. So before that, just some background. So Hernan give a presentation about the protocol, and also we published the protocol in this paper, and you can find most of the details there. So basically, water absorption is about the weight change after cooking. So it's percentage of weight change after cooking. So you can measure 20 times after cooking, 30 times after cooking. So we find the correlation between cooking time. So the cooking time is measured by the fork with experienced person. So how many minutes the roots will be ready after will be ready. So some variety take about 60 minutes. Some good variety take only 15 or 10 or 20 minutes. And then we check the correlation between the cooking time or the optimum cooking time with the water absorption after 30 minutes. So we observed that 30 minutes really give us the best correlation. So in our breeding, we usually use 30 minutes cooking and to measure the water absorption or the weight change. This is the material we use in this study. It was developed by Nielsen and by Dr. Hernan Ceballos. And so we cross the materials with good cooking quality and poor cooking quality. You can see the parents are here. And here are the 5 faucet families, a number of progeny. So in total, we have 237 progeny. And also we have the parents in the field and the check. We plant all the material in Palmyra is in 2020, and we plant them in 2 lots. One is kind of towards is close to the sandy soil. The other is close to the clay soil. And also we plant them in different time. And for each lot, we harvest them twice. So it's 11 months and 12 months after planting. So in total, we have 4 environments. So it's 1, 2, 3, 4, corresponding to 2 lots and 2 harvesting time. So for in each lot, we only have 1 replication, and we have parents and check, and 4 to 6 plants in 1 row. And we measure dry matter, water absorption, and HCN. So dry matter, water absorption, we have 2 and 4 for harvest, but for HCN, we only have 1 environment for today. And also we have some plant type, vigor, disease, insect resistant data, but here we are only present dry matter and water absorption. So first we combine the data from the 4 environment, and we calculate the heritability. So this is dry matter from near. The heritability is is pretty good. It's really aligned well with what we observed before. And so here is 20 minutes water absorption and 30 minutes cooking of water absorption. So in general, the heritability is moderate, is 0.5. And here, the scatter plots is just to show you is the 2 lots, P1 and D1, the first harvest. So you can see the kind of have a feeling about the correlation between the 2 environments. Here is the water absorption in the two lots in the first harvest. So, and in this population, so our conclusion is for water absorption, we believe is at least it have moderate heritability in terms of genetic pattern and the environment pattern, right? And here is just to give you some, just to realize again the correlation among the environment. And I will briefly show. So here is lot one first harvest, lot one the second harvest, and the second lot first harvest, the second lot the second harvest, and then the population mean. So we call it blob. So we have these five data sets, and we can see the correlation between each other, and especially if we compare the two harvests in the same lot is here. So this is D1, the two harvest, and for the P1, so this is so these two data sets is the same field, the two harvest. We see so pretty good correlation. And so on the others, it's mainly showed the correlation between the lot. So this is one lot, this is another lot. So here is show the correlation between lot, is also showed the correlation between lot. So in general, we saw pretty good correlation coefficient among these four environments. And so this is about water absorption. Is a little bit lower. It's also aligned well with the moderate heritability, and the structure of the figure is quite similar with the previous ones. And you can see the correlation between progeny. So this is mainly the population, and also the correlation between the parents. So since we only have several parents, so this is not accurate enough. But for the progeny, we believe is give us a pretty good idea about the correlation among those environments. And then next, we want to look at the family effect among for dry matter and water absorption. So first, to look at the dry matter. Y-axis is dry matter, is the population mean, and Y is the different families. So you can see any bar with two clone names. So that's one family. So this is the two parents. And you can see, and this is the performance of the two parents. And you can see that for dry matter is in general is in the middle, one parent, the other parent, and also for this family, the two parents are neighbors here. And the third group is 25, and this 36. So in terms of dry matter, so this one, the parents is a little bit better. And for dry matter for this group, for the fifth families, yeah. So in general, we can see for dry matter is there's not much transgressive segregation. So it's mainly located between the two. The performance of the progeny is mainly located between the two parents. And similar trends we also observed for water absorption. And you can see for the parents, we are thinking the water absorption is around 10 or larger than 10. Then we can consider they are good. They have good green quality. And you can see there are several parents is pretty low. There is almost no water absorption during cooking. And we compare the distribution between the progeny and the parents, and the conclusion is also quite similar. In general, the segregation within the 4-SIP family is in will be loaded in from the poor performance parents to the good performance parents. Is in general is in the middle, and there is very little transgressive segregation. So that's is much better than one parent. So that's kind of give us idea in breeding. So if we want to improve the co-equality, so we would be better to cross the two parents with good co-equality. And if we cross the good ones with poor ones, in general, you will have very little chance to reach the level of the good parents. And then we look at the correlation between dry matter and water absorption. So we know dry matter is very easy to measure. And in our previous study, when we established the protocol, we used 36 clones, and we didn't observe correlation between dry matter and water absorption. But in this specific population, we saw significant correlation between dry matter and water absorption. But if we look close at the distribution, and we are thinking, so usually the clones with low dry matter, they also have low water absorption. But once the dry matter reach to certain level, so the correlation will be much smaller. So we make the conclusion that if the clones with too low dry matter, it will not have good co-equality. But once it reach a certain level, the dry matter with co-equality might not have very strong relationship. So in terms of the working plan for this year, 2022, so we have in 2021 we planted all these clones in one location. Again, so we are going to have 2 harvests, and we are going to collect the dry matter, water absorption, and HCM. And now we are genotyping the population. So after we have the phenotype data from 6 environments and the genotype data, we are going to do genetic analysis, identify the major QTL, and understand the population, understand the traits much better. And in the same time, for breeding, we are selling the good quality material. So this is something we are planning to do hybrid cassava breeding using inbred progenitor or semi-inbred progenitor. So the material with good quality will provide good germplasm for trait integration in the future. So this is something we are doing now to do selfing, and also we are doing conventional recurrent selection to cross the clones with good quality together, and also cross the innate good quality progenitor with the treated donors, or with the good quality material from the gene bank. Try to again try to improve the quality much better. Yeah. So I think that's all what I plan to present about water absorption. And again, here I really appreciate the support from the quality lab led by Thierry Chan, and also John is mainly manage all the activities in the quality lab. And also thanks for the breeding team members. They manage the trial, collect, harvest the roots to supply to the quality lab. And thank Hernan Ceballos for the supervision and for the ideas. And also thanks the RD Food and NextGen support for these activities. Now I'm open to questions. Thank you very much.
Thank you very much. Thank you very much, Xiaofei, for this presentation, and congratulations actually to the teams, to the food science and the breeding teams together, partly from NextGen and RTB Foods, for this very interesting work, which will surely pave the way for similar work on other RTB crops, because several other teams are actually developing protocols to measure water absorption in RTB foods. So it will surely be duplicated on, for example, yam and sweet potato, hopefully. And we are looking forward to know more about the feasibility of identification of genetic markers. So congratulations. So we are now ready for questions from the audience. You can either raise your hand or write your question in the chat box. I don't see, do we have questions in the chat box yet? Yes, okay. So there's a first question from, I think, Toyin. Was your water absorption measured in dry basis or wet basis?
Yes, it's wet basis. So we, if you can see the equation, so it's the fresh weight before cooking and the fresh weight after cooking. So that's I believe that's wet base. Yeah.
Question from Suzanne. It looks as though only a small portion of the variability in water absorption is explained by total dry matter content. Any thoughts on what other constituents or factors could be influencing water absorption during cooking?
Yeah, so that's good comments, and we also believe for dry matter is not the driver for the water absorption. So there is some hypothesis or assumption that pectin might be the major factor for affecting water absorption. So Thierry, the work package 2 is working together, try to figure that out. So we for some population, the work package 2 is doing, so they measure water absorption. They're also going to measure the pectin composition and pectin amount. So from there, I believe we are going to test that hypothesis and to see whether pectin is a major factor there. Yeah.
Thierry, you raised your hand. Do you want to add something to Xiaofei?
Yeah, maybe I can comment on the starch and pectins. So we don't have all the proof yet, but the story I think is happening in when we boil cassava is that we have the starch, a carbohydrate, a very hydrophilic carbohydrate, which is like a sink who tries to absorb as much water as possible. And because we have a lot of starch, whatever the dry matter content, whatever the starch content, it will always be as much water as possible will be absorbed by the sample. So the starch is like the accelerator trying to absorb the water. And next to that, we have the pectins, which form a network in the cell walls around the starch, which is contained in the cells. And those pectins act as a barrier to the water absorption. So the pectins are like the brake to water absorption. And what will control the water absorption and the amounts, the percentage of water that we see absorbed by the root is going to be the thickness of the pectin, the density of the pectin network to act as this barrier that will block more or less the absorption of water. So it's really this model of starch accelerating, trying to get the water into the piece of cassava, and pectins slowing down that absorption. It's an interplay between the two. we need to gather more data, especially on pectins, to prove that hypothesis.
Yeah,
and I encourage anyone, any team who can help and try to explore this would be helpful to accelerate the process and get to a conclusion as soon as possible. Thanks.
Santiago, I see you raising your hand. Is it just linked to what Thierry just said directly?
Yeah, it's linked to the water absorption. Yeah, it's only a suggestion, but we are focused on the chemical part of it. So the starch and pectins, and maybe the porosity of each sample maybe have a role also. So from a more physical point of view, the porosity of each sample can just change the water absorption too. It's only a suggestion.
Thank you.
Thanks.
Thank you, Santiago. Question from Didier. Did you measure conductivity of the raw material? And if yes, is there any correlation with water absorption at 30 minutes?
Yeah, my understanding, the conductivity is not something we measured. Maybe Thierry, yeah, I didn't hear anything about conductivity. Maybe Thierry can comment. Yeah.
Yeah, we had planned to, and then the lockdown happened, and we lost the samples.
Okay, so is there any evidence the conductivity can have effect on clean quality?
So far we don't have any information.
Yeah, okay.
There is a little challenge that because we are boiling, so there is evaporation of water, and we don't manage to control that. So between different pots and different samples, there may be more or less evaporation, which will have an effect on the conductivity, not because of the samples, but because of the water evaporation. So there is some methodological development to do to control the water evaporation.
A question from Enoch. What is the heritability for water absorption at each site and across sites?
Yeah, thank you very much. So this, this is something I need to clarify. So the heritability we report here is cross site, also is the heritability cross environment. And since in each site we only have one replications, and it's not easy to calculate the accurate heritability. So after we have the genotypic data, so combine the genotypic data, we are going to get the relationship that will help us to calculate the heritability within each site. So that's something we are going to do later. Yeah. Thank you.
Thank you. Akim is actually not asking a question precisely on water absorption, but he said that you've showed that you've been measuring cyanide in only one environment. Can you please clarify? Does it mean that G/E does not have any effect on cyanide?
No, we believe there is also large G/E for cyanide, but due to the workload and due to the COVID, we are not able to complete all the samples. So that's why we only have one environment. So this year we are going to have 2 harvests. So we are trying to get more SCN data. So that's also different environment. We will see the variation. Yeah.
Great, thank you. A comment from Wasiu. He says, I think it would be nice to evaluate the correlation between water absorption, so cooking ability or cooking time of the cooked cassava roots, and the sensory and instrumental texture attributes. So it is more for Thierry, I think. Correlation between water absorption and sensory or texture, instrumental texture.
Yeah, we have the data and we are working on that. And also we are doing the harvests at the moment, and we are accumulating more data.
Yeah, so we have another population with I believe there are 36 clones with different queen quality. So we are collecting water absorption data, we are collecting texture data, we are collecting the sensory data. So using that population, we can try to answer this question. And so the same population we published in the paper here. So this is yeah. So we use the same population to evaluate the sensory and texture. So that's so again, as Thierry mentioned, that's something we are doing now. So
yes, question from Dominique is asking, given the heritability values that has been just shown, which strategy could be proposed for the future to accelerate selection in breeding program?
We are using water absorption to screen early stage material. So in the breeding, in the breeding pipeline, there are several stages, and in each stage we have smaller population size. So now after the second year selection, we harvested the second year selections, and we send the sample to the lab, and they are going to collect the water absorption data. And water absorption is a medium throughput method. Every day we can process at least 80 samples. So using that water absorption data, we already started to trim down our population size to only select the best ones for the next stage analysis. So this is help a lot for the breeding program in terms of labor using, field using, and to manage the cost. So this is what we are currently doing. And in terms of for the future, so we are now there is another experiment is going. So we try to compare the water absorption between environment. For example, we compare the north coast of Colombia, so that's lowland, tropic, and subhumid region, and compare with the trials in Palmyra, so that's above 1,000 meters above the sea level. So we want to see the difference among this environment. So that will give us an idea how the data from Palmyra can predict the north coast. So this is something we are trying to figure it out. If using the data from Palmyra from the CIAT the campus, that will give us a lot of convenience in terms of accessibility of the root material. In terms of NIRS, let me see. I think Thierry has been working with NIRS. As far as my understanding, there's no pretty good correlation. So we try to, rather than using the NIR prediction to rank the material, so we try to using NIR prediction to separate the material, to separate the good one with the poor one. Then that's something we are exploring. And now I don't have the result how NIR works in terms of predict the water absorption. Another thing I want to mention, so we are using quality spec is a portable NIR. So Thierry got pretty good result in terms of quality spec predict the dry matter predict beta carotene. And so we probably in the future, we can bring that quality spec in the field to measure the root piece and to get the prediction value for dry matter, water absorption, and maybe HCN in the future. But for water absorption, yeah, it is still not that easy to measure using NIR. Yeah.
Thierry, do you want to say something regarding NIR's predictions?
Yeah, no, I think Xiaofei has summarized the situations for water absorption. We can classify into two groups, like slow cooking and fast cooking and long cooking varieties, but it's not perfect yet. The percentage of mistakes is still relatively high, so it's still difficult to discriminate only by years between the good cooking quality that we keep and the bad cooking quality that we will reject. The environment has a strong effect as well. When we do the analysis with the harvests of just 1 year, it's quite good. But when we start to mix data sets from different years, we are losing the quality of the classification.
Thank you, Thierry.
Thank you.
Thank you. Any other question or comment?
Yes, Eglantine.
Yes, please, Gerard.
I am Gerard. Thank you very much. I just wanted to thank Xiaofei for his presentation and ask if this study can be conducted on another RTB food crops such as sweet potato or plantain.
Yeah, I think other crops is also testing how water absorption works. So this might be also depend on crops. It works very well in cassava. Yeah, so I don't have much idea the progress in other crops. Maybe Eglantine or Dominic, you know the activities. But
yam, there is an experiment on yam and also on sweet potato at the moment.
Thank you, Dominique. Thank you.
So nobody's working on boiled plantain yet. So Gerard.
No, I am working on boiled plantain, you know.
So please go ahead. Go ahead with water absorption.
I'm following some work on it, and I think Thierry knows that I'm working on it, and very soon we'll have the results.
Very good.
In ITA, Michael Ladezo can have very good correlation between quality and water absorption in yam.
Yeah.
You know that Mariam is also doing some water absorption in sweet potato in Uganda.
So yes, we are looking.
Hello, I have a question.
To your results. Yes.
Hello?
Yes, please.
Okay, thank you very much, Xiaofei. Once again, the reason why I asked which basis you measured water absorption is because usually I think the moisture content of the genotypes may vary with harvest periods and storage and some other factors. So don't you think this may affect how much your material can absorb if you measure on wet basis?
Yeah, so in terms of cassava, so for boiled cassava, we farmers or customers always cook fresh root. So this is exactly the same thing we are doing in the lab. So we also cook the fresh root and try to mimic the situation. So in terms of the water in how to say the moisture or the water, the dry matter content or the moisture in the roots. So we saw some correlation, but it is not very strong, especially once the moisture of the fresh roots or the dry matter content of the fresh roots reach to some level. So the correlation is not that dramatic. So I'm not quite sure which perspective do you have. So, but for cassava, in terms of protocol or throughput, we are thinking the fresh roots is good sample to work with. But if we have to dry the sample to measure the water absorption of the dry sample, that's take a lot of effort. The throughput might be not good enough in terms of measurement. Yeah, that's my understanding. I don't know, Thierry, maybe you have something to add. I saw your hands up.
Yeah, we have little or nearly no correlation between dry matter and water absorption. In the case of cassava, in other roots and tubers, it may not be the same. But in the case of cassava, we have very little correlation. So we could express the water absorption on a dry basis and try to see any effect, but I don't expect much, because it's not really dry matter that is controlling and driving water absorption, but more the pectins and cell wall materials.
Okay, thank you very much.
Any last comment or last question before we close the webinar? No? Okay, so congratulations again, Xiaofei and Thierry together, because it is actually a collaboration between the two teams. Thank you very much, and we are looking forward to more applications on other RTB crops, indeed, and looking forward also to know whether there is a possibility to identify genetic markers for water absorption. So congratulations.